Hey everyone, and welcome to The Explainer. Today, we're diving into a genuinely mind-bending idea. What if biological systems are essentially highly complex computers? We're going to look at the building blocks of life through a totally different lens today, not as squishy random biology, but as rigorous mathematical computation.
Really think about it. Are the cells in your body actually running complex computer programs right now? Like when you get a paper cut, how do your cells know exactly how to divide and heal it? And maybe more importantly, how do they know when to stop? It looks like biology to us, but deep down, it's information processing. It's literal code.
Now, this fascinating science comes directly from a pretty dense 2006 text called Transactions on Computational Systems Biology, VI. It gathers these absolutely groundbreaking papers that treat biological networks and cell membranes as pure computational structures. So, we're going to break down two of the most fascinating studies from this work to see exactly how biology calculates.
Here's our quick agenda for today. First, we'll look at the biological computer. Second, shuffling the yeast network. Third, cellular bandwidth and signals. Fourth, the math of membranes. And finally, we'll wrap up with a Turing-powerful cell.
Okay, let's jump right into Section 1, the biological computer.
And moving into Section 2, shuffling the yeast network. Let's see what that actually means.
So, led by researcher Pierre-Yves Bourguignon, a team set out to map the operations inside a yeast cell. They wanted to see if its apparent chaos actually hides a really refined architecture. Now, the cell relies heavily on two completely different systems. First, there's the protein-protein interaction, or PPI network. You can think of this as the physical hardware, literally proteins bumping into each other in an undirected graph to form complexes. Second is the transcriptional regulation interaction, or TRI network. And this operates exactly like software. It's highly directed, specifically telling genes when to turn on and off. So the big question for the researchers was, how do these two totally distinct networks interface to actually keep the cell alive?
To figure out just how special this cellular setup really is, the researchers used a mathematical technique called shuffling. Think of it kind of like rearranging a city's connecting highways while keeping the internal neighborhood streets completely intact. They deliberately kept the PPI hardware network and the TRI software network exactly the same internally, but they randomly rewired the equator, that specific interface where those two systems meet. By creating these randomized alternatives, they basically established a mathematical baseline so they could compare it against the real biological network.
Well, the results were pretty stark. When those connections were randomized, the overall network actually lost about 4% of its connected pairs. This proves the real yeast cell actively maximizes its biconnectivity. Basically, it ensures massive, highly efficient bandwidth so signals can successfully flow back and forth between the physical proteins and the regulatory genes. It really wants to be as connected as possible. But here's the counterintuitive part. Despite maximizing these connections, the real biological graph actually maintains longer average distances between individual nodes compared to the randomized versions.
Which brings us to Section 3, cellular bandwidth and signals. Let's make sense of this. Why on earth would a cell maximize connections but deliberately choose longer pathways? Well, it's all about avoiding anonymous data. See, if you use a highly compact, star-shaped network, every single signal routes quickly through a central hub. It's incredibly fast, sure, but the ultimate receiver has absolutely no idea which specific outer node originally sent the signal. The origin is completely anonymized by that hub. A linear chain network, on the other hand, takes more steps. It takes longer to traverse, but the path itself guarantees that the receiver knows exactly where the specific signal came from.
As the paper states, the heterogeneous network is likely to result from a trade-off between causality and signal integration. In biology, a specific signal is literally a matter of life or death. If your cell receives a stress signal from its environment, it urgently needs to know exactly what kind of stress and from where, not just some generic panic alert that tells it nothing about how to actually respond. The heterogeneous network of a real yeast cell perfectly calculates this super delicate trade-off. It sacrifices a tiny bit of speed and compactness to guarantee absolute pinpoint accuracy and causality for every single vital message.
All right, moving on to Section 4, the math of membranes.
Now we're shifting to the micro level. Researchers Nadia Bussi and Roberto Guerrieri built a rigorous mathematical model to understand cellular mechanics. They based it on endocytosis, which is just the natural biological process of a cell engulfing external material. Now, real endocytosis can be pretty messy, right? A cell might engulf an arbitrary amount of fluid or debris. But in their strict mathematical model,
They defined a very specific, isolated action called phago, where a cell membrane perfectly swallows exactly one other external membrane, nesting it entirely inside itself.
To complete this little cellular toolkit, we have pino, short for pinocytosis. This is essentially the membrane folding inward and pinching off to create a small, empty bubble inside itself. So it's engulfing zero external membranes, just drinking in its surroundings. Alongside exocytosis, which is the reverse action of spitting a membrane back out, these highly specific, highly controllable actions form the precise syntax of an entirely new biological math language.
The researchers then pitted two different mathematical models against each other. In the PET model, driven by phago, exo, and pino, when one membrane swallows another, the structural nesting gets deeper, exactly like a Russian nesting doll. You can just keep adding layers inside of layers. Conversely, the MBD model, which stands for mate, bud, drip, relies purely on membranes fusing together or dripping apart. The really crucial difference here is that in the MBD model, the total depth of the nesting never actually increases. It just spreads out horizontally.
And finally, Section 5. A Turing powerful cell. This is where it gets crazy. So, what actually happens when we push the nesting nature of that PEP model to its mathematical limits? Well, it turns out a membrane swallowing another perfectly mimics a computer adding a 1 to a register. The researchers proved you can actually encode a random access machine, which is a foundational model in computer science, using just cell membranes. You represent a basic number n by nesting 2n plus 1 membranes. To add 1 to your count, a membrane simply performs a phago action to make the structure deeper. To subtract 1, it performs an exo action to spit a layer out. It is literal, functional, biological arithmetic happening at the microscopic scale.
And here is the absolute bombshell. Because the PET model can infinitely increase its nesting depth through swallowing, it is Turing powerful. That means a simple cell membrane can theoretically run the exact same computational logic as the computer or phone you're using right now. It can compute absolutely anything that is mathematically computable. The MBD model, since it completely lacks that nesting depth, is strictly limited in its power, and its termination is always decidable. But the PET model? It holds the boundless computational potential of a universal machine.
From the vast, meticulously routed networks of yeast proteins actively prioritizing clear, pinpoint signals, all the way down to the Turing-powerful geometry of microscopic membranes swallowing each other, biology isn't just surviving, it's computing. Your biological network is effectively your body's own private internet, constantly crunching data just to keep you alive. And if life itself is a computer at its most fundamental level, the only question left is, how soon until we learn to write our own code for it? Thanks so much for joining me for this explainer. I'll see you in the next one.